All right, in the lipid section, this is our fats, oils, and waxes portion of the chapter. Keep in mind this section is not in the textbook. That just means that the textbook's polymer chapter doesn't cover it, and that's because it simply isn't a polymer. Different from proteins and carbohydrates that came before it, you don't have a repeating unit here. You do have pieces that make a large molecule, which is why it's important to cover this, because we're talking about the other three major biological molecules anyway. But just notice that these are going to be exclusively in the notes and not found in the textbook when you cover them. So as we look at lipids, what's most important here is to realize that they have functional groups too. So very similar to polyesters that we saw in the condensation portion of a previous video, is these guys have acids and alcohols that make esters. And esters aren't that polar. They do have some carbon-oxygen bonds that are polar, but that's a far cry from the rest of the molecule that is superbly nonpolar with long-chain hydrocarbons that are going to hate water. So as a whole, lipids are pretty water-insoluble. And that's because a majority of their molecule is nonpolar. And that's because it's hydrocarbon-based. It is an ester linkage, and lucky for us, they don't change this in biochemistry or anything else. It's still called an ester linkage, so it's not like the amides going to peptides or the ether-like linkages of carbohydrates that we just saw in the previous video going to glycosides. Here it's an ester, and they call it an ester, and they don't change it, which is nice. Less to remember. Uniquely here, what we find is most of our cooking oils and fats, and some of the waxes that you may not think of chewing on a candle, but a lot of waxes are in things like chocolate. A lot of those are lipid-based. Also, a lot of our vitamins that we need for our body are lipid-based, so they're nonpolar. There's a handful of vitamins that are water-soluble, and those are the ones that are missing in this list. A fun way to remember the ones that are water-insoluble is to remember all dogs eat kibble. A's, D's, E's, and K's are water-insoluble, meaning that they're fat-soluble, meaning that they are nonpolar. And the ones that are missing, like the vitamin B's and C's, those are the ones that are water-soluble. So when you take a big vitamin supplement like these, you can imagine that very little of this material can actually be useful in the body because your water is, your body's mostly water. And because water is so polar, you can only get so much of these vitamins to dissolve and to be used before they're basically filtered out from the liver, from the kidneys, and leave the body. Another feature of this chapter is steroids. This is all of our hormones in our body, and we'll see those. Those are also big and nonpolar. Of all the groups, it's these that are nonpolar that have difficulty in our body finding, use, finding a place, because the rest of our body is polar. We do revisit the idea that in these fats, oils, and waxes, we have saturated and unsaturated hydrocarbons. Saturated hydrocarbons are the ones that are probably the worst to have. That's because they're the ones that are easiest to store, because they're the easiest to stack based on their shape being so symmetrical and so even. Introducing some unsaturated double bonds just means that there's going to be a mismatch in that kind of zigzag. Every once in a while, you're going to have a double bond that's a little bit shorter than those single bonds, and that's going to make them a little bit less easy to stack. And if they can't stack, that just means they're not going to be solids at room temperature like bacon grease or the fat on steak. Traditionally, they're going to be more liquid-like and more oil-like instead of more, less fat-like. Most lipids have a triglyceride kind of structure. The reason we use triglyceride is tri just means 3 fatty acids. The glyc part in the middle is because it comes from glycerol, which is a large alcohol structure. You can imagine here, very similar to what we've seen in a previous condensation processes, is when you have alcohols and acids, here's the water that's going to leave. So in making fats, oils, and waxes, we produced 3 water molecules. That also means that if we're going to take this molecule and digest it, we're going to need 3 water molecules to go back in and break them apart. Unfortunately, that's the problem. These alcohols and these acids are superpolar because of their OHs. So when you form them and produce water, we lose that polarity because now our ester isn't that polar. This chain over here is so long and nonpolar that it's going to really hate water. So the ability for water to come back to these molecules is pretty limited, and that's the problem with their digestion. A lot of people struggle with, breaking down fats is because the body doesn't have a reason to attract to them. So you can imagine that in order to break these apart, you're going to have these left and no other energy source available. So only after a tremendous amount of exercise can you exhaust your body's sugar content. A lot of its glycogen, which is its, animal starch, is going to be used up first. And then only when all sources of easy energy is taken up, then can we go in and say, all right, all that we have left is fat, let's start burning it because we're out of sugar. And the reality is, in most cases, that doesn't happen on a regular basis. So you store your fat for those rainy days of, starving. And oftentimes, that doesn't happen. Definitely not often enough. The only way to make these a little bit easier to digest is with the help of proteins called enzymes. There are lipases that can attract to these nonpolar tails a little bit and digest them. Also, the help of bile salts can help break them down and ionize them, because if they're ionized on one of their portions, so if one of those acids were to fall off and make an ion, then now we have some water solubility to be easier to digest further. So it's very important to realize that these are the toughest to digest. So the ones you want to watch out for in your diet the most, because they're going to be stored, they're not going to be digested and reused very quickly, which is why they tend to stack on your, face cheeks and butt cheeks eventually. There's 3 major types. The first one was a fat. Fats are typically from animals. These are the ones around bacon and pork chops and pork steaks and, ribeye steaks and those kinds of things. So that white, fatty kind of structure is saturated. So when we eat it, it's going to still be saturated, and it's going to stack on us just like it stacked on those previous animals. And you can imagine the reason that it stacks so nicely is if when you look at the structure at the bottom, it shows that very perfectly patterned and organized, kind of bond line structures. If we were to draw them without the hydrogens, they stack easily. And if they stack easily, they're going to stack easily on our bodies and our fat cells, which is also nonpolar. If we switch gears to oils, what's going to happen is now we're going to have some double bonds. And these double bonds can be both cis and trans. So you might hear the idea of trans fats. Trans is an alkene that has opposite endings. So trans looks like this as a double bond, and then cis looks like this as a double bond. So you may hear trans versus cis fats. Trans going to have that same relative orientation. So you notice these zigzags here. If you have a trans alkene, it's in the same kind of pattern. However, if you have a cis fat and one of these were a double bond, it would have its zigzag going in the opposite direction. So these cis alkenes, they're actually better for digestion because they're going to break up that pattern of stacking because of their kind of U shape. Trans fats are just as bad as saturated fats, and that's because they are going to maintain that same kind of overall shape. So shape, polarity, can't tell you enough. It's, crucially important in your body and how it functions. When we say the word wax, most people think of candles, and that's great, but they do put a lot of wax into things like chocolate. This holds its shape, it gives it its shine, it resists it from melting so quickly, but unfortunately it's using, instead of glycerol, a long-chain alcohol, so this is the worst of the worst. At least with a triglyceride, that glycerol portion isn't very big, and it's close to the polarity, so it's easier for enzymes to attract it and to break it apart and digest it. The problem with a wax is it's got huge nonpolar portions on both sides. That's going to give it nonpolar protection in the middle, and it's going to be even harder to digest, probably the worst to try to digest. So if you eat a lot of chocolate, you tend to gain a lot of weight, and this is why. And it's all because of Hershey wants to put their stamp on their chocolate, and in order for that stamp to stay and not get melted and disappear, they're going to have to put a lot of wax in there to keep it holding its shape. The more traditional waxes that we probably heard about are things like plant waxes, so the carnauba waxes that you might put on your car. There are plenty of soy waxes you can use in candles, or different waxes and materials that can be used in things like lotion, or even things like waterproofing on paper. Those are also types of waxes, just not the type that we typically eat. There's other animal waxes like beeswax that are used in a lot of makeup. There's things like beef tallow that are used for emergency fuels, used for leather conditioner, leather kind of things like that would soften a baseball glove or a pair of boots. And there's plenty of other chapsticks and even perfumes. Kind of weird to think that perfume, a lot of the molecules that help it be in the aerosol kind of format comes from sperm whale intestinal lubricant. So pretty crazy that sperm whale intestinal lubricant is used for perfume that you spray on your body. So there you go. Sometimes it's better not knowing, I guess. Kind of a hybrid category of lipids is soap. Soap is what happens when you treat lipids with a strong base like sodium hydroxide. So if you were to throw in some Drano to some vegetable oil, you could make your own homemade soap in a very crude fashion. What's nice about soap is you have this nonpolar tail. If you have this nonpolar tail here, it's all hydrocarbon based. And if all of this is nonpolar, it's going to love the oil. And in this portion over here is very polar because it has a -1 charge. It's ionic. And if it's ionic, it's going to love water. So this molecule has the best of both worlds. It loves water on one side, but it loves oil on the other. And we use this for our clothing, for our hands, for showering and so forth. So very unique because it's using something nonpolar, but being broken apart, no longer in its triglyceride structure. Now it has some polarity on the end so that it can attract to water and be rinsed away. So soap is a hybrid kind of polar molecule because due to the reaction of that oil, no longer is it completely nonpolar. It's now superpolar because it's ionic on one side, on its polar head, so to speak. Another feature of lipids is phospholipids. Phospholipids are used to make your cell membranes. So when you get to biology or physiology class, every cell in our body has a membrane, and they're using these lipids. So in addition to needing fats for energy, we can transform some of those into phospholipids to make our cell walls, and we constantly need new cells in our body. So what's unique here is they use the same feature that they had for soap, but they do this to allow us to have this non-polar inside layer and this polar outside and inside layer. So that separation of polarity allows for discrepancies on who can get in and out of the cell. Because in order to go through the cell wall, you have to be polar on the outside and be attracted to the cell. You have to find the little gates, which are the active transport proteins in many cases that can allow you in. Because the rest of the cell wall is completely nonpolar, and there's very little things in your body that are completely nonpolar that could get past the polar surface to get into that, kind of inner workings that's nonpolar, and then eventually work your way through the other polar layer that's on the inside with all your organelles. So very important that our body has that kind of two rows of defense of polar, nonpolar, polar, because that really regulates what can go in and out of a cell, and that's very important for cell life and cell function. The last part of this lipid category is the steroids, and steroids are all the things that give us our ability to reproduce. So you can imagine if you're a male and testosterone, it puts hair on your chest, it deepens your voice, it helps promote the growth of your reproductive organs. And the opposite is true for the female gender. at least biologically, progesterone going to produce breasts that can feed, the children. It's also going to help develop the reproductive organs to be able to create those children. So it's kind of unique that you can mix and match these hormones. You can give the hormones to the opposite biological sex and kind of reverse those roles. But it's all molecules that determine those kinds of features in our body. And depending on which ones are in a higher concentration, that can determine your hair growth, that can determine your breast growth, it can determine your, your deep voice versus, high-pitched voice. All of those kinds of ideas, your muscle mass, those are determined by these kind of lipid hormones that we call steroids. Notice all of them end in O and E, at least testosterone and progesterone, and that's because they have that ketone portion of their molecule in that lower kind of left side. Cholesterol is something that a lot of people may have trouble dealing with, and notice it's very similar to the hormones. So our body sometimes has difficulty knowing which is which because they are, pretty identical in their structure, having that 6-6-6-5 kind of ring structure. So in our last installment of this chapter, we're going to talk about DNA, and that's probably the most complicated of the bunch. So we'll catch you on that video later.